Hello, Sangeeta here. Today's topic for discussion is integration of metabolism. So as we all know, human beings derive their energy by oxidation of foodstuffs, that is nutrients. We need a constant supply of energy which is required for all cells to survive and function normally. The balance between the need and the mobilization of stored energy is called metabolic homeostasis or calorie homeostasis. So this metabolism, which serves the following purposes, like it is specifically to yield chemical energy which is obtained from the degradation of energy rich nutrients. These food materials are converted into macromolecules such as proteins, nucleic acid and polysaccharides. The metabolic pathways which take place with the help of sequential enzyme system, and these pathways which are regulated at three levels. A regulation through the action of allosteric enzymes, hormonal regulation and regulation at the DNA level, that is gene level or transcriptional level, where the concentration of enzyme is changed by regulation at the level of synthesis of the enzyme. Now regarding the types of metabolic pathways which occurs in human body, there are mainly three types of metabolic pathways. Catabolic pathways, that means the degradative pathways, where usually the energy rich complex molecules will be degraded into smaller molecules, in which the energy released during this process is trapped as chemical energy in the form of usually as ATP. And anabolic pathways, that is synthetic pathways, the cells synthesize complex molecules from simple precursors. This needs usually energy, whereas catabolic pathway which synthesizes energy, where anabolic pathways which requires energy for the biosynthetic pathways. But sometimes in body amphibolic pathways are also seen, like at crossroads of metabolism, where both anabolic and the catabolic pathways are linked. The best example, TCA cycle. Now comes to the stages or phases of metabolism, which occurs in three stages: primary metabolism, secondary and tertiary metabolism. So during the primary metabolism, the foodstuffs undergoing digestion in the GIT tract and converts the macromolecules into smaller one. For example, proteins are digested into amino acid. Similarly, carbohydrates are digested into glucose. This is called primary metabolism. And after the metabolism, these products will be getting absorbed, catabolized to smaller components, and ultimately which will be oxidized to carbon dioxide. So in this process, usually the coenzymes NADH or FADH2 are generated, and this process is known as secondary or intermediary metabolism. And finally, these reduced equivalents like NADH, FADH2, etc. will enter into the ETC, that is electron transport chain or respiratory chain, where energy is released. This is the tertiary metabolism or internal respiration, or which is also known as the cellular respiration, without which human beings cannot be survived. Now comes to the major fuels of the human body. Those are glucose, fatty acids, amino acids and ketone bodies. So these are ultimately which we are getting through the food digestion and absorption, and ketone body we are not getting through the food, but which we are getting through the specific condition where acetyl coenzyme is converted into ketone bodies. So this table which explaining the amount of various nutrients in human body in different stages like starvation, walking and marathon. Okay, so for example, fat A reserve of 15,000 grams fat is available in human body. This is sufficient to maintain the energy during starvation for 34 days. And if you are walking, which will be utilized by 11 days and marathon by 3 days. Similarly, muscle glycogen, 350 gram muscle glycogen stores are there in body, which is sufficient to meet the energy needs for 14 hours, 5 hours by walking, 17 minutes for marathon. Like that. Body protein, 6,000 gram, which is sufficient to meet it for 15 days during starvation, and if it is by walking 5 days it will get depleted. Then if we are doing marathon, which will deplete it by 1.3 days. So these are the major metabolic pathways which we have already learned. Like under the heading of carbohydrate metabolism, we have learned major and minor pathways like glycogenesis, glycogenolysis, then glycolysis, gluconeogenesis, etc. That means glycogen synthesis, breakdown of glycogen, oxidation of glucose to pyruvic acid or lactate, then synthesis of glucose from non-carbohydrate substrates. Minor pathways like HMB shunt, especially for the synthesis of NADPH, then uronic acid pathway for the production of glucuronic acid, then lactose synthesis, etc. Under the heading of fat metabolism, we have learned the lipogenesis, fatty acid synthesis, lipolysis, that is breakdown of lipid, then the oxidation of fatty acid, then ketone body synthesis and ketone body breakdown, that is ketolysis. Then under protein metabolism, we have learned the catabolism of amino acid, that is proteolysis, then these amino acid oxidation, then urea synthesis, and also regarding the protein synthesis. So this is a major metabolic pathways interconnection, or how these pathways are getting integrated. Like the carbohydrate, then fatty acid and amino acid. So after the catabolism or after the oxidation, which will be converted into pyruvic acid, that is glucose converted into pyruvic acid in aerobic condition, fatty acid which is oxidized to acetyl coenzyme A, then amino acid after the catabolism, the carbon skeleton of ketogenic amino acid converted into acetyl coenzyme A, or it can be even converted into pyruvic acid, and pyruvate can be oxidized to acetyl CoA. And acetyl CoA is finally the final ultimate product of the metabolism of all these nutrients, will enter into TCA cycle for the complete oxidation, and which will be converted into carbon dioxide, water and ATP. ATP not directly produced here. The coenzymes NADH, FADH2, GTP, etc. are synthesized to TCA. Now the organs concerned with metabolic integration are mainly liver, skeletal muscle, adipose tissue, gut and kidney. These are the organs mainly associated with the metabolic integration. As far as liver is concerned, which is the central metabolic clearing house of body, the site of entry of all foods following ingestion, that is, we all know that after the digestion, absorption through the portal circulation, these biomolecules will be entering into liver where the final metabolism occurs. So this liver maintains circulating nutrient levels through various metabolic pathways. Now comes to the skeletal muscle. This is a major energy consumer. Takes up and utilizes more glucose, ketone bodies, fatty acids, etc. Where glycolysis is a preferred ATP production pathway in exercising muscle. Now adipose tissue, which is widely distributed throughout the body, an average 70 kg human possesses about 15 kg of fat tissue. The mass of adipose tissue, which contains 135,000 kilocalories of energy, which is sufficient to sustain life up to 35 days without any food intake. So this much storage of fatty acid or fat in the form of triacylglycerol in the adipose tissue is sufficient enough to live the life for 35 days without any food intake. Now brain it makes up only about 2% of the total body mass but uses the 20% of total body energy consumption. Glucose is a predominant fuel used by the brain, but in prolonged starvation, brain can utilize ketone bodies as fuel. From glucose, it switches to ketone body as a fuel. Brain does not store any fuel stores. So this figure which explains the utilization of energy by nutrients in fed state. For example, glucose will be utilized by brain, but in starvation, ketone bodies as utilized as a major source of energy in the brain. A brain switches from glucose to ketone bodies. The major organs solely dependent on glucose for energy are brain, then RBC, retina, renal medulla, testis. So these organs which require a constant supply of glucose which is essential for these organs to maintain the energy level. So the starve feed cycle, well-fed state 1 to 4 hours after food, that is known as a well-fed state. Early fasting between 4 hours and 16 hours, that is known as early fasting. Fasting is known as between 16 to 48 hours. Starvation means 2 to 3 days of deprivation of food. And prolonged starvation means greater than 5 days without any food if the person is continuing, that is known as prolonged starvation. So the well-fed state, what is the meaning? During well-fed state, absorption of glucose from gut occurs. So this causes increase in blood glucose. And increase in blood glucose which stimulate the beta cells of pancreas and which release the secretion of insulin. The fate of fat in the fed state, dietary fat which is absorbed, which is re-esterified in the intestinal mucosal cells and which used to pack into chylomicrons, which delivers free fatty acid and glycerol to the peripheral tissue. In adipocytes, again a re-esterification of this free fatty acid occurs and which will be stored in the form of triacylglycerol. So this is showing the inter-organ transport of amino acid after taking food. So we have seen the how the glucose and fat absorption and utilization during inter-organ transport occurs or in postprandial condition. Here the amino acid, especially from muscle, alanine is transported in the fed state, but during fasting state, from muscle the alanine will be transported to the liver. Then glutamine transported to kidney and valine will transported to brain. So this is explaining the postprandial condition. So postprandial condition, from liver, alanine will be transaminated and it will be transported to muscle. So this is metabolism in well-fed state. So after taking food, glucose will be absorbed and amino acid and fat in the form of fatty acid will be absorbed, and excess glucose will be converted into glycogen and stored in liver and muscle, and glucose can be directly utilized by the brain cells and other peripheral tissues. Then fatty acid can be converted into very low density lipoprotein and can be moved to the adipose tissue. The metabolic events which occurs during early fasting between 4 to 16 hours, fuel stops from the gut because we are not consuming food. So it leads to decrease in blood sugar level. The insulin level slows down. Blood sugar level maintained by hepatic glycogenolysis. There will decreased glycogen synthesis and decreased lipid synthesis. Glucose paired for brain. The available glucose will be utilized only by the brain cells that occurs during the early fasting hours. So this is a figure which explains the metabolism in fasting, what all we have discussed. Opposite of that happened during postprandial state occurs. From muscle, the amino acid alanine is transported to blood and which will be utilized for the gluconeogenesis. Through gluconeogenesis, glucose will be synthesized in liver. The newly synthesized glucose will be transported to brain cell. And glycogen which is present in the liver will be broken down to glucose and also can be utilized by brain cells. And adipose tissue, hydrolysis of TG occurs and liberation of free fatty acid and which enters into liver cells, and this can be utilized by in the form of ketone bodies by peripheral tissues. So this happens where during there is no food, that is during starvation. So this again which explaining the interorgan transport of amino acid during fasting condition. Opposite that of postprandial condition. So during fasting condition, alanine transported to liver, utilized for the gluconeogenesis. Glutamine transported to kidney to utilized for the acid-base balance and also for the other mechanism. And valine will be transported to brain for the synthesis of ketone bodies. Now comes to the metabolic adaptations during starvation. During fasting, for first 18 hours, blood glucose is maintained by hepatic glycogenolysis. Remember, 18 hours the glycogen storage can utilize to maintain the blood glucose level. In the second stage, gluconeogenesis comes up. Even before the glycogen storage getting depleted, gluconeogenesis is accelerated. The amino acid released from muscle form the major substrate for the gluconeogenesis, and the glucose-alanine cycle will be getting activated. Then the branch chain amino acid liberated by protein catabolism are utilized by the muscles to give energy. Brain completely take up the glucogenic valine from the bloodstream for the ketone body production. Third stage, lipolysis occurs, in which the skeletal muscle, heart, kidney, etc. will shut down their glucose utilization and will depend mainly on the fatty acid for the energy needs in the form of ketone bodies. Fourth stage, acidosis occurs, the production of ketone bodies which leading to metabolic acidosis. The pH falls and hyperventilation occurs as a compensatory mechanism. Fifth stage, if the person still continues with the prolonged starvation, death from starvation occurs. Metabolic acidosis, dehydration, etc. will be aggravated unless corrected efficiently, that leads to the person's death. A normal person has fuel reserves to live up to 45 to 60 days without any food. Further, not consuming, death is sure. So hormonal regulation during well-fed state is mainly done with insulin. During starvation, it is regulated by glucagon and catecholamines. And starvation, the decrease of regulation which is done with thyroid hormones and decreased metabolic rate. So during starvation, the basal metabolic rate will be decreased in body and the thyroid hormones also decreased. Metabolic changes seen in diabetes mellitus almost similar to starvation. What all we have discussed, the stage 1 to stage 5 occurs in diabetes mellitus. In spite of adequate glucose, every tissue switches over to catabolic roles. Metabolism stick to starve phase of cycle, resulting in life-threatening conditions. Here, it is not because the lack of food, but the glucose is not efficiently utilized due to insulin deficiency. So this figure which explaining the overall interrelationship of metabolic pathways and end products of secondary metabolism. So you can see protein after the metabolism It is finally converted into ammonia, which will be eliminated in the form of urea, and also the nitrogen pool, which will be utilized for the synthesis of various things. The carbon skeleton of the amino acid can enter into formation of pyruvic acid, formation of acetyl-CoA, formation of citric acid intermediates, etc. Then carbohydrates, which undergoing various metabolic reaction, depends on the body needs either synthesis of glycogen, well-fed condition, breakdown of glycogen during fasting condition. Similarly, glycolysis during well-fed condition, gluconeogenesis during fasting condition. And pyruvic acid during well-fed condition can convert into acetyl-CoA. Similarly, lipids, well-fed condition it undergoing lipogenesis, fasting condition which undergoing breakdown of fatty acid to form acetyl-CoA. So in well-fed condition, the end product of all the metabolism is acetyl coenzyme or other T-cell cycle intermediates, which undergoing T-cell cycle as a complete oxidation pathway to produce carbon dioxide and water, and the metabolic intermediates in the form of coenzymes will enter into ETC cycle for the complete production of ATP. So this is all about the integration of metabolism. Thank you.